Namaskar. Welcome to Introduction to Microorganisms. We are continuing with our module on bacteriology. We have already finished introduction, size and shape, prokaryotic appendages, prokaryotic growth, prokaryotic reproduction, and domain bacteria. Now we are continuing with the second domain of the microorganisms in the living three domain classification, that is domain Archaea. Archaebacteria are chemically distinct from other bacteria in several ways. The cell walls, cell membranes, and ribosomal RNA are different from those of other bacteria. They have no peptidoglycan. They are extremophiles, and the prefix Archaea was given to them because they are considered to be ancient, because they are considered to be of the time period when Earth had no place for normal condition growing organisms like usual bacteria. These extremophilic unusual prokaryotes are hence named as Archaea or Archaebacteria. Archaebacteria live in conditions similar to the era when life first appeared and began to evolve on planet Earth. Archaea share certain traits with bacteria and other traits with eukaryotes as expected. Archaea have specific types like methanogens, which produce methane and can be used in sewage for degradation of carbon sources and further production of methane. Thermophiles or thermoacidophiles or hyperthermophiles, they are growing on acidic hot water springs, as can be seen here. These are the extremophilic organisms. Halophiles, which grow in extreme salt content, where they are growing at a salt content which is about to get crystallized. Archaea, as prokaryotic cells, have cytoplasmic membrane which has ether-linked lipid to glycerol. They have 70S ribosomes, typical of prokaryotes, and they have 16S rRNA. They differ from bacteria in the absence of peptidoglycan, and they are similar to eukaryotes on the basis of presence of histone-like proteins which are associated with the DNA. Staying true to the prokaryotic cell, they do not have true nucleus, but they have nuclei in the cytoplasm, and they lack membrane-bound organelles. Like bacteria, Archaea are unicellular, but they have extreme shape differences, just like bacteria. They can be pleomorphic, they can be square, they can be any other shape, they can be long, slender, or they can be the same shape of a bacillus. When compared with respect to membrane composition, Archaea show high protein content like bacteria. Archaea show sulfolipids, glycolipids, non-polar isoprenoid lipids, and phospholipids, which are seen usually in both eukarya and eubacteria. They have straight chain lipid structure similar to that of bacteria, while eukaryotes have branched lipid structure. Archaea have ether-linked, di and tetraether-linked lipid linkage, while bacteria and eukarya show ester-linked lipids In Archaea and bacteria, sterols are absent, which is present in the cell membrane of eukaryotes. Cell wall. Archaea have variable cell wall composition, which means they do not have same type of cell wall in all the types of Archaea that we have been studying. In Methanobacterium, glycans or sugars and peptides can be the cell wall, while in the Methanosarcina, non-sulfated polysaccharides are cell walls. In Halococcus species, sulfated polysaccharides are cell walls, while in Halobacterium, negatively charged acidic amino acids which counteract positive charges of high sodium content in the environment. They also have cell lysis in initial concentration less than 15%. That's why they are extremophiles, growing in extreme conditions of salt. Methanomicrobium and Methanococcus, their cell wall is made up of exclusively protein subunits. As can be seen in this image, gram-positive bacterial cell wall has a cell membrane or cytoplasmic membrane with teichoic acid and lipoteichoic acid and peptidoglycan on the outer stage, while in gram-negatives, the cytoplasmic membrane has an outer membrane and a periplasmic space with small layer of peptidoglycan. In Archaea, different types of cell walls are present. In some, pseudopeptidoglycan is present with heteropolysaccharide cross linkages, while in Methanococcus and Halobacterium, S layer, an extra layer of lipid or an extra membranous layer is present on the surface of the cytoplasmic membrane. Chromosome, ribosomes, RNA polymerase. With respect to these, the Archaea show both eukaryotic and prokaryotic, specifically bacterial traits. They have similar DNA replication machinery as eukaryotes, while their genome is single circular chromosome like bacteria. They have histones like eukaryotes, while they show operons like bacteria. They have nucleosomes like structures, while they do not have introns like bacteria. They have transcription machinery with RNA polymerase, transcription factor 2 beta, and data binding proteins like eukaryotes, while like prokaryotes, bacterial type membrane transport systems are there. Like eukaryotes, they also exhibit initiation factors in translation machinery and ribosomal proteins, elongation factors, and which are poisoned by diphtheria toxin. But as bacteria or eubacteria, they have many metabolic processes for energy production, nitrogen fixation, and polysaccharide synthesis. But till now, the studies have concluded that all the Archaea are chemotrophic, that is, they grow on chemicals, and they show enormous phenotypic diversity. As can be seen here, they are chemotrophic, that is chemolithotrophs, or they can be considered as chemolithoautotrophs or chemolithoheterotrophs. They can be chemoorganoautotrophs or chemoorganoheterotrophs. When we consider bacterial metabolism diversity, many Archaea are extremophiles, which are growing at the extreme conditions. As can be seen, highest temperature resistance is by an Archaea, that is Pyrolobus fumari at 90 degrees Celsius minimum and 106 degrees Celsius optimum with 103 degrees Celsius survival rate. Low pH Picrophilus oshime, which can grow at pH as low as minus 0.06 and can grow up to maximum of 4 pH. High pH alkali, that is Natronobacterium gregori, with highest of up to 12 pH. Pressures and high salt content is also high with respect to Archaea. Thermophilic Archaea, they are extreme thermophiles, survive at 350 degrees Celsius under tremendous pressure. Optimal operating temperature is just over 110 degrees Celsius. They survive anaerobically at 250 degrees Celsius, and these cells could have survived on a young Earth under conditions that are thought to be uninhabitable by all known life forms. Paralobus fumari is the organochemolithotroph which can survive in extremely high temperatures. Archaea as methanogens which form methane. These are obligate anaerobes. They are present in marine freshwater sediments, deep soils and intestinal tracts of animals like us. They use hydrogen for energy, carbon dioxide from carbon source, and produce methane. They are associated with heterotrophic eubacteria and protozoa, but these are the most diverse group of Archaea. These are extreme thermophiles, moderate thermophiles and mesophiles also. Methanogens morphology can be cocci, rod or spirillum. This is the Volta experiment where the gas or methane produced at the base or benthic area of the small water body like a lake can be collected in an inverted funnel in the form of methane gas, and the gas can be used for igniting or as a biogas. Halophilic Archaea, formed in salt flats and evaporation ponds. Color of these areas are pinkish red because of the presence of Archaea. They can't live in salt concentrations below 10%. That is usually where bacteria grow. They are bright red carotenoid pigment which protects the cells from intense solar radiation. Bacteriorhodopsin, which is produced by these Archaea, use sunlight for energy. These produce their own ATP using this pigment. Using this pigment, ATP is produced by chemiosmosis on the membrane. As can be seen in this image, these reddish or pinkish areas are the evaporation ponds for salt production, which are full of Archaea or halophiles. This is the water content where high Archaea can be seen. Thank you. Here the module on bacteriology is finished. In our next module, we will discuss kingdom Mycota, which includes fungi. Thank you.